All right, so what I want to look at now is after we have taken our pyruvate in that pyruvate oxidation phase that we talked about previously and converted it into acetyl-CoA, so acetyl coenzyme A, remember that's 2 carbons and we made 2 of those because we had 2 pyruvates. What do we do with that acetyl-CoA, right? So essentially what we're doing is we are going to put it into this loop. We're going to put it into this cycle in which acetyl-CoA is going to combine with this other molecule to form a 6-carbon molecule, and then systematically this 6-carbon molecule is going to get broken down into a 5-carbon molecule, which is then going to get broken down into a 4-carbon molecule, and then it's going to reform the same one that we started with. Okay, so that's why this is called a cycle. Acetyl-CoA is going to join with something called oxaloacetate, and we'll talk about what that is. We'll look at it in the next slide. That's going to join with the acetyl-CoA to form something called citrate, which is 6 carbons, and then this citrate is going to get systematically broken down into a 5-carbon molecule, and then into a 4-carbon molecule until it gets reformed into oxaloacetate, and then it starts over. So that's why this is called a cycle, because it is occurring. It's occurring in which you reform the same molecule that you started with, right? So each acetyl-CoA enters this cycle and causes one revolution of the cycle, if you will. So that's for one acetyl-CoA. And remember, we have 2 of those because we had 2 pyruvates, and the 2 pyruvates came from 1 glucose. So you have to be careful in thinking about what are the products of this process. Are you talking about a single rotation, a single cycle? If so, that's only half a glucose, right? Because one cycle is 1 acetyl-CoA, which corresponds to 1 pyruvate, which corresponds to half the glucose. So for a single cycle, this is what we're making: NADH, FADH2, and 1 single ATP molecule. That's one cycle. If you want to say what's the products per glucose, then you would just multiply those by 2. So 6 NADH, 2 FADH2s, and 1 ATP, right? So we've talked about this NADH before. NADH is something called nicotinamide adenine dinucleotide. It is the reduced form of NAD+, so it has a hydrogen added to it. And again, it's carrying electrons. What we'll do in our last phase, called the electron transport chain, oxidative phosphorylation, is we'll use these NADHs and the FADH2 that I'll mention in a second. We're going to use those to essentially pump hydrogen ions across a membrane to create a gradient, and then use that gradient to make ATP, right? So these are coenzymes, right? So NADH, FADH2 stands for flavin adenine dinucleotide, right? And it's doing a very similar role to NADH. It's carrying electrons, take them to the electron transport chain, and help us make ATP, right? So we make a little bit of actual ATP here. We make some NADH, we make some FADH2, and we also make some waste products. So we make some CO2, right? So when we started as a 6-carbon molecule with that citrate, and we end up with 4-carbon molecule, the oxaloacetate, the 2 carbons that are lost as carbon dioxide. Okay. So this is a little bit more accurate representation of the citric acid cycle, or the Kreb cycle. It's called the citric acid cycle because it starts here. The first molecule that's formed when you combine this oxaloacetate and acetyl-CoA, the first molecule that's formed is citrate here, right? Notice all of these enzymes, they all end in ACE, right? ASE. So you know it's an enzyme. And you go through until you reform, you go through this loop until you reform oxaloacetate. And then that recombines with acetyl-CoA to form citrate. You go through this process and you reform oxaloacetate. And it combines with a new acetyl-CoA, forms citrate, and this process continues and continues and continues so long as we have oxygen, right? So remember, this is an aerobic process, right? This is an aerobic process. So for this course, I am not worried about you knowing the specific names of each of these intermediate molecules. So don't worry about that too much. However, the ones that I would like you to at least know are, obviously know acetyl-CoA, because we've talked about that previously. Know what it combines with, this oxaloacetate, and then know that it forms something called citrate, right? Those are kind of the main things that I want you to think about here. Okay, but again, we're going through this process. We are making some NADHs, we're making some FADH2s, we're making some byproduct, we're making some water and carbon dioxide. But the big thing are those NADHs and FADH2s. Okay, those are going to be extremely useful and necessary when we get to our electron transport chain. Okay, so again, this cycle is occurring once per acetyl-CoA, which means once per pyruvate, which means twice per glucose. Okay, so again, be careful if you're talking about the products. Are you talking about the products that are produced per cycle, per loop, or are you talking about the products that are formed per glucose, right? Because there's a different answer there. Okay, so in reality, the real Krebs cycle. So this isn't the real Krebs cycle. That I should have mentioned the term Krebs cycle. That's named after the scientist who described this process initially, named Hans Krebs. So if you discover and describe a process like this, you get to name it after yourself, apparently, right? So Hans Krebs, that's where the term Krebs cycle comes in. So Krebs cycle, citric acid cycle, right? In reality, the real cycle is this, right? You learn the Krebs cycle and then you forget the Krebs cycle, right? So you learn it, you learn the enzymes, you learn the products, you learn the various things that go into it, what's produced, and then you forget it. And then you have to learn it again, and then you forget it again, right? So this is the real Krebs cycle. Okay, so let's stop here, and what we'll pick up with in essentially what will be our last part is looking at something called the electron transport chain, and we'll see how we use those NADHs and those FADH2s that we've been producing. We're going to see how we actually use those to produce lots and lots of ATP.